Hi, I'm Mark Feinberg. I'm a cardiologist, vascular biologist, and director of cardiovascular RNA biology research at Brigham and Women's Hospital and associate professor of medicine at Harvard Medical School in Boston. I'd like to share with you some of our ongoing studies in this talk entitled LincRNA Regulation of Vascular Senescence and Atherosclerosis. As you know, accumulating studies over the years demonstrate that atherosclerosis is a chronic inflammatory disease driven in part by activation of endothelial cells that orchestrate the expression of adhesion molecules and chemokines and the recruitment of inflammatory leukocytes, including monocytes and T cells. Lipid-laden macrophages from the necrotic core of the plaque and traditional risk factors such as cholesterol, high blood pressure, and diabetes accelerate chronic inflammation and atherosclerosis. And as you know, statins reduce risk through both lipid-lowering effects and anti-inflammatory mechanisms. The inflammatory hypothesis has been recently validated in the CANTOS trial that used an IL-1 beta antagonist to reduce cardiovascular events in secondary prevention. We wanted to know if there may be new pathways that could be leveraged to impact atherosclerosis. My lab has focused on the role of non-coding RNAs, both microRNAs and long non-coding RNAs, also known as lincRNAs, in atherosclerosis. These non-coding RNAs are derived from about 80% or so of the genome that is transcriptionally functional. In contrast to microRNAs, lincRNAs are much longer, defined as greater than 200 nucleotides in length. Most are capped, polyadenylated, and spliced. And while they are poorly conserved, those that are tend to have a cell or tissue-specific expression. In contrast to just a few thousand microRNAs, there are over 50,000 human lincRNAs, and their function remain poorly understood. These lincRNAs can interact with RNA, DNA, and protein, providing new levels of regulation that we need to think about in atherosclerosis. While the role of lincRNAs are being identified in different aspects of cardiovascular disease, as shown on the left, a direct role in the vessel wall remains poorly understood. So we set out to gain a better understanding of the role of lincRNAs in atherosclerosis. Our general aims are to identify intimal lincRNAs that are dynamically regulated with lesion progression, uncover their molecular mechanisms, and to determine their translational relevance. So here's the experimental setup. In the upper left, you can see we harvested aortic intima for RNA sequencing using an intima peeling technique from LDLR deficient mice. These are atheroprone mice, placed first on a high cholesterol diet for 0, 2, or 12 weeks, reflecting a progression phase. And then we told these mice it's time to go on a low-fat diet, and we placed these mice on a normal chow diet for an additional 6 weeks, reflecting a regression phase. From RNA sequencing, amongst the top hits was a lincRNA termed SNHG12, also known as small nucleolar host gene 12, which decreases with atherosclerotic progression and increases with regression. We found that this lincRNA is conserved across species, and interestingly, its expression is reduced in atheroprone regions of the vasculature and is reduced in non-atherosclerotic mice with increasing age. So we wish to first characterize this lincRNA. In the upper left, you can see that this is an endothelial cell enriched lincRNA expressed higher in the aortic intima than the media or bone marrow mononuclear cells. It does not encode a protein or peptide and is polyadenylated, all consistent with the lincRNA. And then we found that it is specifically expressed in the nuclear fraction of endothelial cells by real-time PCR and cell fractionation, and by fluorescent in situ hybridization. So to explore the function of SNHG12, we used inhibitors called gapmers, which are antisense oligonucleotides capable of reducing lincRNAs in the nucleus using an RNase H-dependent mechanism, which you can see here on the right. We also wish to verify that the knockdown of SNHG12 had no effect on its neighboring genes, which it did not, as shown on the right below. So to explore its role in the pathogenesis of atherosclerosis, we optimized a delivery protocol for tail vein injection of these gapmer inhibitors to SNHG12 or a scrambled control or a standard 12 weeks of high cholesterol diet in LDLR deficient mice. And as shown on the right, this achieved approximately 50% knockdown of the expression of this link RNA, SNHG12, in the aortic intima. There was no effect in the media, and there was a modest knockdown in peripheral blood mononuclear cells. Surprisingly, this knockdown triggered a profound 240% increase in atherosclerosis in the aortic sinus and 170% increase in athero in the descending aorta as quantified using oil red O staining. Upon characterization of these lesions, we noticed something unusual. There was no difference in NF-kappa B staining, reflecting inflammation in the vessel wall. There was no differences in the circulating lipid profiles in the upper right, and there was no difference in the accumulation of leukocyte subsets, such as macrophages or T cells, in these lesions. So what is driving the increased atherosclerosis? And perhaps it's independent of our usual suspects of lipids or inflammation. And at this point, we really didn't know the mechanism, so we went fishing for the interacting partner of this link RNA with the aim of informing a better understanding of the biology. To do this, we performed biotin-labeled link RNA pull-down studies in combination with mass spec and endothelial nuclear lysates and identified a protein termed DNA protein kinase, or DNA-dependent protein kinase, or DNA-PK for short. And this is one of the three major arms involved in the DNA damage response and implicated in cell senescence and apoptosis. And here on the left, you can see validation studies showing that this link RNA, SNHG12, pulls down DNA PK specifically, but not other members of the DNA damage response. And on the right, we performed domain mutation studies and found that domain 4 of this link RNA to be critical for this interaction. And below on the left, using reverse immunoprecipitation pull-downs of DNA PK, we found that DNA PK is capable of interacting with link RNA SNHG12, highlighting that this is a bona fide interaction between this link RNA and this protein termed DNA PK. As shown here, DNA PK is a major regulator of DNA repair in response to DNA double-strand breaks, induced, for example, by either extrinsic stimuli, such as irradiation, or intrinsic stimuli, such as reactive oxygen species, cytokines, or even lipids. And on the right, you should know that DNA PK interacts with two chaperone proteins called Q70 or Q80 to mediate DNA repair through a process called non-homologous end joining. So now, armed with a potential mechanism, we turn back to the accelerated atherosclerotic lesions and found that there was indeed a twofold increase in lesional DNA damage in the vascular endothelium, as shown here in the upper right, using a marker gamma H2AX, which marks for DNA double-strand breaks. And in addition, loss of SNHG12 endothelial cells attenuated binding of DNA PK to its chaperone proteins Q70 and Q80, reducing DNA repair and non-homologous end joining. To more definitively address whether SNHG12 regulates DNA repair, we stably transduce human umbilical vein endothelial cells with a DNA repair reporter called PDRR, in which the intron-encoded endonuclease SCI1 cuts the integrated reporter to generate double-strand breaks. Repair through non-homologous end joining leads to expression of green fluorescent protein. And indeed, on the right-hand part of this figure, you can see that lentiviral overexpression of this link RNA SNHG12 promoted non-homologous end joining, as indicated by a 2.5-fold increase in GFP positive cells compared to controls. To further explore the effect of SNHG12 on DNA double-strand breaks in endothelial cells, we performed complementary approaches using loss and gain of function studies. In the upper left in B, you can see that SNHG12 deficiency predisposes endothelial cells to DNA damage in the presence of gamma irradiation, a very potent form of double-strand breaks. In C, SNHG12 deficiency increases gamma H2AX, again, a definitive marker of DNA double-strand breaks, in the presence and absence of hydrogen peroxide, a well-known physiological inducer of reactive oxygen species and double strand breaks. Whereas in D, SNHD12 overexpression protects against DNA damage with reduced phosphorylation of gamma H2AX expression in the presence of hydrogen peroxide. And finally, in E in the lower left, you can see that SNHD12 deficiency increased DNA damage using the COMET assay. It's a complementary assay measuring the tail lengths of individual cells as a marker of DNA damage. And furthermore, this increase was blocked in the presence of knocking down its interacting protein, DNA-PK. Taken together, these findings indicate one, that SNHD12 deficiency triggers DNA damage in the form of double strand breaks. Two, SNHD12 overexpression is protective and reduces DNA damage. And three, SNHD12 is dependent on DNA-PK for mediating these effects. Because non-homologous end joining can affect either apoptosis or senescence, we first examined for any effects on lesional apoptosis in atherosclerotic mice treated with SNHD12 inhibitor, gap mirrors, or control gap mirrors. As shown here, there was no significant effect on cleaved caspase 3 as a marker for apoptosis. Further examination of atherosclerotic lesions, however, revealed marked increases in senescent markers P16, P21, and P27, and increased acellular areas, also known as plaque necrosis in mice, that received inhibitors to SNHD12 compared to controls, highlighting increased senescence in these lesions. And consistent with these findings, knockdown of lincRNA, SNHD12, and endothelial cells triggered that senescent marker SA beta gal in endothelial cells. So we still wanted to explain the increased deposition of lipids in these lesions in these SNHD12 knockdown mice. One of the hallmarks of the senescent vascular endothelium is that it becomes permeable, especially to LDL, in a process termed LDL transcytosis, in which LDL gains access to the subendothelial space. And using TIRF, or total internal reflection fluoroscopy, to quantify fluorescently labeled LDL for its ability to get into the subendothelial space, you can see in this video a small amount of LDL flickering in the left, in the bottom left, in the control group. However, on the right, there's a near doubling of LDL in the presence of SNHD12 knockdown, demonstrating that SNHD12 deficiency promotes LDL transcytosis. To examine the role of SNHD12 on endothelial cell permeability in arteries in vivo, we used a cone-shaped polyethylene constrictive cuff that was placed on the left common carotid artery to mimic disturbed flow in lesion-prone areas. Flow perturbation was measured 24 hours after surgery by Evans blue extravasation. And as you can see on the left, the downstream blue area of the constrictive cuff was more elongated in longitudinal cross-sections of the left common carotid artery after SNHD12 inhibition compared to the negative control. And conversely, on the right, administration of SNHD12 RNA by intravenous injection reduced downstream Evans blue extravasation. These data highlight that SNHD12 dynamically regulates endothelial cell permeability in the vessel wall in vivo. So if DNA damage and senescence were the main mechanisms driving the accelerated atherosclerosis, we wanted to know if we can rescue the development of atherosclerosis by SNHD12 gain of function studies. To address this, we synthesized SNHD12 RNA by T7 in vitro transcription coupled with 2O-methylation and capping. In vivo delivery of SNHD12 RNA or control RNA in ApoE atheroprone mice revealed significant accumulation in the aortic intima, but not in the media or peripheral blood mononuclear cells. And on the bottom, you can see that SNHD12 overexpression reduced the atherosclerotic lesion formation and DNA damage, specifically for double strand breaks using the marker gamma H2AX in the vascular endothelium. So how does SNHD12 regulation of DNA damage response impact mitochondrial damage, which typically goes hand in hand. Using RNA sequencing and network pathway enrichment studies, we identified one of the downstream pathways in response to SNHD12 inhibition is indeed P53, which can impact mitochondrial bioenergetics. To address this, we performed oxygen consumption studies using seahorse and endothelial cells. And as shown here, SNHD12 inhibition impaired oxygen consumption rates, including ATP production and altered NAD-NADH ratios in endothelial cells. One of the known compounds that can potentially impact DNA damage is nicotinamide riboside, or NR for short, which can activate NAD, a known deacetylase that reduces ROS and DNA damage, and has been linked to a well-known longevity signaling pathway. So we assessed whether NR could rescue the effects of SNHD12 in endothelial cells. And here you can see that NR effectively improved oxygen consumption rates and improved the NAD-NADH ratio. So we reasoned that if nicotinamide riboside rescued DNA damage and downstream effects in endothelial cells in cells in a dish, perhaps it might rescue the effects in vivo. So we repeated the atherosclerotic studies with SNHD12 or control gap mirror inhibitors in atherosclerotic LDL deficient mice, but in the presence now of NR in the food. And here you can see the results. You can see that NR effectively blocked the acceleration of atherosclerosis. It blocked DNA damage as quantified using phosphorylation of gamma H2AX for double strand breaks. And it blocked vascular senescent markers, P16, P21, and P27. And finally, it reduced acellular areas, also known as plaque necrosis. So we think these results may have some clinical or translational relevance. Expression of link RNA SNHD12 is reduced in atherosclerotic lesions, not only in mice, but also in human carotid atherectomy specimens, as shown here on the left, and associated at the same time with increased markers of DNA damage, gamma H2AX, and senescent markers such as P16 and P21. This appears to be conserved across species. Expression of SNHD12 in the bottom left is also reduced in pig atherosclerotic specimens that also express a higher level of senescent markers, as shown on the right. So our study builds upon several emerging studies that indicate that cellular senescence may be an independent pathway promoting atherosclerosis. Indeed, atherosclerosis is associated with alterations in cell proliferation potential, markers of cell senescence, increased cell death, DNA damage, and telomere shortening. All are hallmarks of vascular senescence. And as shown below, a report a few years ago showed that deletion of P16 senescent cells using genetically modified mice was indeed sufficient to reduce atherosclerotic lesion formation in these mice. So in summary, this link RNA SNHD12 is a DNA damage responsive link RNA. SNHD12 acts as a stabilizer of DNA PK interaction with Q70, Q80. Deficiency of SNHD12 increases DNA damage, impairs non-homologous end joining, and increases senescence. This results in phenotypic consequences on endothelial cell permeability to LDL. And we learned that reduced expression of SNHD12 increases atherosclerosis independent of the lipid profile or lesional leukocyte accumulation via the DNA damage and vascular senescence. We found that nicotinamide riboside can rescue the DNA damage, vascular senescence, and accelerated atherosclerosis induced by SNHD12 deficiency. And that delivery of SNHD12 RNA to overexpress this link RNA reduced plaque burden, DNA damage response, and senescence, both in endothelial cells in vitro and in vivo. And that these data have some translational relevance for being conserved across mice, pigs, and human subjects. So finally, I'd like to thank the duo in my laboratory who performed the work, Dr. Stefan Hamig and Dr. Dafan Yang, for all their efforts on carrying out this link RNA project, as well as other members of my lab, my collaborators, and funding sources. And if you're interested in a position and joining our non-coding RNA program, you can reach out to me directly via the email address below. So thank you for joining me on this presentation today. Thank you for your attention.